It's the 8th of May, 2020. This is the RheumNow podcast, and I am Dr. Jack Cush, executive editor of RheumNow.com. Peter Nash loves it when I say that. This week, the title of the show is The Beat Goes On. Of course, I'm talking about the COVID crisis, that strange new reality that is unfamiliar, if not unnerving, and just doesn't seem to want to stop. But yet, we have lots of new news and journal reports that's probably worth talking about. I'll start off with some general rheumatology. We've talked about anakinra and its use in pseudo-gout before. EULAR and ACR had reports on that. Well, I think they materialized in a meta-analysis recently where they looked at 74 patients who were treated with anakinra for refractory CPPD, or a few, and that was about 85%, maybe about 20% who had other contraindications to other therapies. And in this cohort, anakinra was effective in 81% of those with acute CPPD or gout, and in 43% of those with chronic disease. That's sort of new data. The good news is very well tolerated, adverse events in only 4.1%, something to consider beyond steroids or colchicine in people with acute CPPD or pseudo-gout. There's only one drug on the market that's approved for non-radiographic axial spondyloarthritis, and that is, right, certolizumab or Cimzia. Well, there are other drugs that have been studying this and looking at this, and that includes Cosentyx or secukinumab, and this past week, the EMA has approved Cosentyx for its use in active non-radiographic axial spondyloarthritis based on the results of their phase 3 trial, the PREVENT trial. It is not yet approved for non-radiographic axial SpA in the US. I would expect it to be so in the months to come. There's an article that we missed. I think it was reported in JAMA a few weeks ago about cigarette smoking in ANCA-positive associated vasculitis. Really interesting study. John Stone and coworkers looked at 473 ANCA-associated vasculitis patients, compared them to about 1,400 controls, and did basically smoking questionnaires. They showed that the ANCA-associated vasculitis patients were more likely to be former smokers or current smokers, with basically having a 1.6 or 2.7-fold increased risk of developing ANCA-associated vasculitis. However, it was only seen for MPO-associated ANCA-associated vasculitis and not with PR3 AAVs. Kind of interesting. So vasculitis being a major issue, let's talk about vasculitis and a COVID patient, specifically about the use of rituximab. I found this as an interesting case report. You remember 3 weeks ago, we said, B cells could play a role in COVID infections, and talked about an Italian case series of 2 patients with A-gamma globulinemia that did better than the 5 or 6 patients who had, what was the other one? They had a common variable immunodeficiency. The common variable immunodeficiency patients did poorly. The A-gamma globulinemic patients looked like they did really, really well. Well, here's a report of a stable GPA patient who's been on rituximab for over a year, received their last dose of rituximab on the 5th of March, and the next day developed symptoms of active COVID infection. Within 4 or 5 days, the patient had progressed, was hospitalized, had high fever, developed respiratory failure, went on mechanical ventilation, was then treated with the usual, onslaught of many drugs, including antiviral therapy, 10 days of BID hydroxychloroquine, and guess what? The patient continued to do well. On day 20, the patient was extubated. At that point, the patient was negative for nasal PCR swabs for active infection, was discharged 9 days later on day 29 and went home. The point of the paper was, 1, you can get COVID infection taking rituximab. 2, this wasn't a death, so I don't think that's a negative story. That might be a positive story in this N of 1 experiment. Again, I think B-cell therapy, you can't really stop it. You might want to avoid it in people who are well controlled if they develop the infection. But again, this patient did fairly well. The World Scleroderma Foundation came out with recommendations for the management of COVID in that population with scleroderma and its variants. It was a long paper with a lot of recommendations, including the usual ones on social distancing and hygiene and whatnot. And I tweeted 5 points that I thought were worth repeating, especially to a scleroderma audience. Number 1, patients with systemic sclerosis could be at risk largely because of their lung disease, and only if they have lung disease, that being interstitial lung disease. So again, there I might be saying extra precautions would be in order for someone with systemic sclerosis and lung disease. Number 2, it's best to continue scleroderma therapies, mainly because you don't want to avoid the therapy and develop a lapse in disease control, which could increase risk. Number 3, scleroderma patients don't need to be tested unless they have evidence of symptoms suggested of this infection. Number 4, ACE and ARB should be continued in these patients. That's been the general guideline for many agencies while they're studying the role of ACE2 receptors and what they mean in doing well or not doing well with COVID. And lastly, number 5, there is no good reason to start or use hydroxychloroquine as a preventative agent in patients with scleroderma. It doesn't work with scleroderma, so I'm not sure why you would even be using it there. Maybe to manage arthritis, that might be possible. Some bad news is coming down the line about the financial consequences of this disorder. The American Hospital Association has projected that in the U.S., there'll be a loss of $202.6 billion between March 1st and July 1st in the United States. That is roughly a loss of about $50 billion a month while we're under siege and under lockdown. When this will change, hard to tell, but that's the projection for right now. An interesting study came out this week from Gut. That's a GI journal where they talked about a limited cohort study of IBD patients, 79 patients, and these 79 patients were reported because they developed the COVID-19 infection. In that population, 46% had pneumonia, 26% were hospitalized, and 8% died. 6% were admitted to the hospital, and what they basically found that were at the outcomes were more likely to be related to active IBD and disease activity and not related to IBD treatment. So again, it was the IBD and or the comorbidities that we would associate with bad outcomes with COVID infection, but it wasn't the therapies that were being used for treatment of inflammatory bowel disease. An interesting report comes from the Journal of Allergy and Clinical Immunology, where a group of French investigators reported on their studies in 26 patients, specifically looking at type 1 interferon immunoprofiling in COVID patients. Type 1 interferons, as you know, are important in innate immune responses and have antiviral properties. They can run amok during adaptive immunity when an excess of type 1 or alpha interferons can lead to a worsening of autoimmune disease and maybe even infections. As we heard from Lenny Calabrese during Grand Rounds on Tuesday night, there seems to be in many patients a deficit of interferon, and that during the later phases when they're doing poorly, there's an overabundance of it. Well, in this particular study of 26 patients who were critically ill, hospitalized in the ICU, and being treated with standards of care, including antiviral therapies or immunotherapies. These patients did not do well. 14 out of 26 were on mechanical ventilation. When they looked at alpha interferon 2A, they showed that there was a rising peak in levels by day 8 to day 10. They peaked, and this sort of coincided with the increase in viral replication. And what they did show was that 20% of their population failed to make a rise in type 1 interferon. Turns out that those were the people who tended to do the worst, have longer ICU stays, worse outcomes, etc. An interesting report from Italy about type 1 interferons playing a role in COVID. So on the website this week, we talked about a number of things related to outcomes in COVID-19. Thursday's report looked at thrombotic complications, really based on a number of reports that have shown up in the literature in New England Journal, in the British Journal of Rheumatology, and in a pathology journal looking at basically vascular outcomes in COVID. And what was reported, and what you can piece together, what I pieced together was, number one, there's a lot of vascular pathology in the poorer outcomes in the late phases of COVID infection, where people die. So in the late phase of COVID infection, people that get to that phase are ones that have usually handled or gone by the viral phase of the illness, and then have gone into a disease magnifying phase driven by an abundance of inflammation. If they're unlucky, they get the cytokine storm syndrome. And there are other patients who are having complications related to thromboses. So in the pathology series, the autopsy series that I quoted, yes, half of them had bronchopneumonia related to viral infection, but there were a lot of patients with pulmonary embolism, pulmonary vasculitis, thrombotic microangiopathy, basically pointing to the role of vascular disease in these poorer outcomes. Researchers at Johns Hopkins are starting to look at the mechanisms underlying the thrombotic microangiopathy seen in COVID patients, and they postulate that it looks like a complement-mediated process, or what Michelle Petrie has called a complementopathy, where there's activation of C5 and downstream effects leading to a lot of complement-mediated damage, including all the things that you see in that terminal phase and the cytokine storm. They have lymphopenia, high LDH, anemia, sometimes microangiopathic anemia. They have organ damage. Again, this is what is commonly seen in HUS and in the catastrophic antiphospholipid syndrome. And anyway, these in this British Journal of Rheumatology report, they postulate that this is a complement-mediated thrombotic microangiopathy, and make a case for complement inhibitors being studied in patients with COVID infection, severe COVID infection. In fact, those trials are going on right now. Also, New England Journal yesterday talked about the presence of patients with the lupus anticoagulant with active COVID infection. 34 patients studied, 31 had evidence of a lupus anticoagulant. This sort of seems to dovetail well with an earlier report in New England Journal looking at the presence of antiphospholipid antibodies, IgA antiphospholipid antibodies, along with glycoprotein 1 antibodies as well in patients who had thrombotic complications and CNS disease. So it is seeming to come together. I think that this is what has led to a lot of centers treating hospitalized COVID patients with anticoagulation, and then there are these trials of complement inhibitors. We need to see more of this going into the future. I had a report about hydroxychloroquine and Qt Prolongation. That comes from JAMA Cardiology. It's a study of 90 hospitalized patients with severe disease. These 90 patients were treated with hydroxychloroquine. There were 53 of them. About half of them were also on azithromycin. Overall, there was a 20% risk of QTc prolongation in those treated with hydroxychloroquine, defined as a greater than 500 millisecond QT interval. This was a little bit more frequent in those who are also taking the azithromycin or those who are taking loop diuretics. I bring this up because there seems to be a lot of concern about cardiovascular outcomes in the antimalarial treated patients. Recognize, number one, these are sick patients with COVID who are hospitalized and have other cardiovascular comorbidities. These are patients who are getting high doses of chloroquine and or hydroxychloroquine, and maybe it is under this circumstance that patients may be at higher risk for arrhythmias or cardiac complications or cardiac death, etc. Turns out a lot of these deaths and a lot of these severe cardiac complications seem to be more linked to the comorbidities and the age of the patient rather than to QTc prolongation. But again, I don't think this is going to change what we do in rheumatology. We're using low doses, we're monitoring, we don't see this. I don't think an EKG is in the near future for any of my patients who are going to be started on hydroxychloroquine. That would be a different story if one of my patients was being treated instead for active COVID infection with current protocols. Yes, monitoring EKGs and QTc intervals seems to be prudent. But in our regular care, I don't think we're going to be changing the cardiac risk profile of what we're doing. Again, this tends to be a relatively rare phenomenon in our RA and lupus patients taking hydroxychloroquine. I had an interesting report, I thought, because I wrote it, on hydroxychloroquine and a lot of the confusion that abounds. You know, I basically said that there seems to be reason for it as to why it might work hypothetically, but in reality, it hasn't worked in other infections, and in reality, it hasn't yet proven itself to work. There's actually a New England Journal report from yesterday showing it didn't harm and it didn't help, and there are several reports showing worse outcomes in early reports. But you can read that if you want to get the lay of the land on hydroxychloroquine and COVID. But I'll end with the report, part of the report that looked at basically supply issues of hydroxychloroquine. So what we do know is that it was a real concern that this was going to go into short supply and our patients with RA and lupus wouldn't be able to get the drug. And there have been some reports of some constraints on finding drug, but it turns out that in the last month that hasn't seemed to be the case. Well, if you look at the FDA website on drug supply issues, there's only one report of a short supply, and that comes from Sun Pharmaceuticals, where they say they have limited availability. If you look at the other source of short supply and shortages, that comes from the AxSpA site. That would be the, I don't know what that stands for, American Society of Hospital Pharmacists, something like that. Number one, they list that there are many suppliers of hydroxychloroquine, including Amneal, Dr. Reddy, Major, Concordia, Mylan, Prasco, Rising, Sandoz, Sun, Teva, and Zydus. Of these, less than half of them have a listing of having a short supply. That it would include Major, Rising, Sun, Teva, and Zydus. And that they say for most of them that they expect to replenish their supply within the next few months. There are a number of reports about stockpiling of hydroxychloroquine. There's a report that 22 states and local governments have reported to stockpile 30 million doses of hydroxychloroquine. In addition to that, FEMA has shipped and stockpiled over 19 million hydroxychloroquine tablets that were distributed to major cities around the United States and to VA centers. So again, I think it looks good. Number one, because of the negative reports about hydroxychloroquine and chloroquine may lessen the fervor to hoard this drug or to take it as prophylaxis, which really nobody should be doing these days. And I think that, again, there is enough supply out there right now to get us through, I think, the bulk of this into the summer. So here are my recommendations about hydroxychloroquine. Number one, don't use hydroxychloroquine or chloroquine to prophylax against the COVID infection. There's no evidence that would work. Don't stop hydroxychloroquine in your patients who are on it chronically. There's no sense in stopping good therapies, safe therapies that are working well in patients just because we're in an environment of hydroxychloroquine craziness. It is okay to start hydroxychloroquine during this era as you normally would. I think that if it's the preferred drug for the condition you're treating, go ahead and use it. I think it makes sense. There's no reason to stop. No, I don't think there's any reason to stop hydroxychloroquine in patients at the doses you use in patients who are actively infected with COVID-19. recognize that these drugs are being studied in many centers around the United States, and if the patient is hospitalized and already taking hydroxychloroquine, they'll be excluded from those clinical trials, but they should likely continue the drug. Overall, there is an exceedingly low risk of cardiotoxicity or cardiovascular death in the doses and the manner in which we use hydroxychloroquine in our patients with rheumatoid arthritis and lupus and other indications in your clinic. I think it's okay to refill hydroxychloroquine as you normally would. My patient wants a 1-month supply, they get a 1-month supply. If they've been getting 3-month supplies, I give them a 3-month supply. I let the local pharmacy be a bit of a gatekeeper, and we've seen some of that, where I write for a 3-month and only 1 month is filled, but there's plenty of refills there. No one so far in my clinic has gone without hydroxychloroquine. And lastly, if you have a patient with suspected, one of your patients in your clinic with RA, lupus, SPA, psoriatic arthritis, whatever, and you suspect them as having COVID-19 or they have confirmed COVID-19, please register that patient with the Global Rheumatology Alliance. The website is rheum-covid.org. They're collecting amazing data. They have over 1,000 patients accrued thus far. In 2 weeks, you'll hear from the guy who started that, Dr. Philip Robinson, and he's going to tell you the update from that registry and what's going on. A lot of interesting videos this week. I have a video coming up very soon with Barry Gruber talking about denosumab and whether you should be stopping that during the COVID era. I have an interesting conversation with 2 practitioners, Danny Ricciardi and Herb Baraf in Maryland, about how they're getting through the current era. And lastly, be sure to tune in every Tuesday night for Tuesday Night Rheumatology, the Grand Round series. Next week, Joan Merrill from the University of Oklahoma is going to talk about what's the difference or what the similarities are between COVID and lupus. Should be an interesting session. Thanks for tuning in. Watch the website for more news. Take care of yourselves.